My name is Will Wan, and I'm a professor in the Department of Biochemistry. The focus of our research is to determine the molecular mechanisms of viral life cycles, and in particular we're interested in negative sense single-stranded RNA viruses such as Ebola virus or rabies virus. So one of the primary tools in our lab is cryo-electron tomography, which allows us to look inside of complex biological environments such as cells or viruses and visualize the individual molecules. So this is still a relatively cutting-edge method, so a lot of our work revolves around developing new hardware and software methods as well as applying these methods to solve new biological problems. Yeah, so my advice to trainees or postdocs entering the field is to really make sure this is something you're interested in, and once you're sure about that, you can pursue your interest. And I think part of that is both being in the lab experimentally, but a large part of being a scientist is everything you do beyond the bench. So those are things like pursuing academic scholarship and learning new ideas and consulting with literature. I think the more freer you can be with your ideas, both with respect to science and even interests outside of science, the wider your kind of tools for assessing problems becomes, and the more creative your research can be. The way I generally view research is that science is a very creative type of occupation. So in running the lab, my goal is to make sure that the people in the lab are as creative as possible and able to kind of pursue whatever their interests are, whatever their ideas may be. So for me, basic research is extremely important. I think some of the greatest discoveries in the last 100 years is from basic research, because nature's been doing this for a very long time, and we don't really understand all the ways in which nature accomplishes its various tasks and various needs. So basic research allows us to freely pursue ideas without the need of generating a product in the end. And by doing so, we have the biggest opportunity to learn new things and advance the field, which eventually finds its way to produce products and different therapeutics that ultimately help people on a more tangible level. But basic research is key to broadening the horizons and really helping to narrow down on new ideas. So I'm known primarily as a structural biologist, and this is a field in which we study the structures of molecular machines and basically figure out how they work from there. My kind of most cited work is probably my work on Ebola virus and determining the structure of its components. Part of what attracted me originally to this field is the interplay between mathematics and physics and chemistry in order to solve biological problems. I guess part of my way of seeing these problems as quantitative problems, and fields such as structural biology or molecular biophysics really allow you to approach biology from those directions. In particular, I found myself interested in viruses because they're very interesting molecular machines that are a bit different than other forms of life in that they require other forms of life and they're actively kind of fighting against something that's actively fighting against them. So the interplay of these different aspects and how they align with one another are interesting things to study. Funding is always a complicated thing. Definitely it's required to not only hire people, but particularly in my field, we use very large instruments, which means that they're very expensive to purchase and operate. So funding there becomes a thing that can be somewhat constraining, but at the same time, I think having a bit of constraint also makes sure that you're pursuing kind of the best ideas and pursuing your research in a way that is most efficient. So originally I did my PhD here at Vanderbilt, and what attracted me then as a graduate student was the very open environment and the ability to really pursue your research interests. And those are the same reasons that brought me back here. The main things there are that the barrier to research here is very low, in that research is generally operated as a very open field and it's not separated by departments or anything like that. So, pursuing research that is outside of my direct expertise is pretty straightforward to do here, with the combination of openness and also the ability to consult both my colleagues and also professional core staff that are better at these methods than I may be. it's always a bit of a jarring transition going between a postdoc and a faculty member, because suddenly there's a lot more responsibility, especially when as a postdoc you don't have to defend a PhD or anything in the end. So it's very much the most open time of your career. But I think part of that transition is something that I was ready for as I was coming up to it, and that it got to the point as a postdoc where I had more ideas and I had time to carry them out. And that's really when I felt ready to transition towards a professor role. So for me, the most important thing is a combination of enthusiasm and tenacity. Even the smartest students will often fail, and if you're doing good research, a lot of it will be failure. And key to being a successful scientist is being able to evaluate your failures and figure out from there what the next step forwards are. Yeah, I mean, that's definitely how science needs to go. You need to, take your hits, maybe take a weekend off and reevaluate things and relax and take your mind off of things. But in the end, you have to just kind of get back on the horse and go at it. I think diversity is a really important part of a research environment. So I did my postdoctoral fellowships abroad in Germany at very international institutions where basically within the lab there was never more than 2 people from the same country. And that also, and the amount of diversity and richness that we have in the United States really kind of emphasized how different backgrounds and different points of view can lead to new ideas and different ways of pursuing the same problem. And I think that's always a very important part of pursuing science, in that creativity and is the most important thing for solving problems. So having a diverse group that is well connected and communicative with one another, I think, is one of the most efficient ways to really solve your problems. Yeah, so I would say that my academic background probably starts at high school, where I received my high school diploma from Stuyvesant High School in New York City, and that was a science and engineering high school, and there, that's kind of really where my interest in science began. And from there, I did my bachelor's degree at Binghamton University as part of the State University of New York system. And I pursued a biochemistry degree there, but my interest also lied heavily in kind of chemistry and the physical aspects of biology. And that's what took me here to Vanderbilt. In particular, I came in through the integrated graduate program, or the interdisciplinary graduate program, the IGP, which provided me the opportunity to both expand my research interest in pursuing different types of rotations, as well as having a broad enough research environment for me to find a lab that really suited me. After completing my PhD here with Gerald Stubbs, I went on to do 2 postdoctoral fellowships abroad. So the first one was at the European Molecular Biology Lab in Heidelberg, Germany. And that's where I started studying viruses and studying cryo-electron tomography. I then moved on and did a second postdoc at the Max Planck Institute for Biochemistry in Martinsried, Germany. And there I continued my studies of cryo-electron tomography into more complicated cellular systems. And after that, I came and joined the faculty here.